This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 518, recorded on November 1st, 2018. I'm Vincent and you are listening to the podcast all about viruses. Today, I am recording at the Penn State University College of Medicine. It's in Hershey, Pennsylvania, where I'm the guest of the Department of Microbiology and Immunology. And before we start talking about science, I want to ask you to please subscribe to my YouTube channel, youtube.com/PROFVRR. We need 25,000 subscribers to be able to apply to YouTube for educational grants. And it's free to subscribe, costs you nothing. So please go there right now on your phone or your computer, youtube.com/PROFVRR. Today I have two faculty members from the department here, and we're going to talk about their careers and their research. And I have picked a relatively new investigator and, shall we say, an experienced investigator. Let's put it that way.
Old, maybe?
No, I wouldn't use the old word. So let me welcome to TWiV from the department, Leslie Parent. Welcome to TWiV.
Thank you very much. Happy to be here. It's a real honor.
And so Leslie is the experienced investigator, and we have a relatively new investigator, Nick Bukovic. Welcome to TWiV.
Thank you. It's a pleasure to be here.
Thanks for both of you for doing this. I know it seems a little scary, but it'll be fine because we're going to talk about you after all, and what better things to talk about. You should know all the answers to these questions. So again, we're at Penn State in Hershey, where there's a lot of chocolate everywhere, wherever
Absolutely.
you go. This is a chocolate town. The town was founded by Milton Hershey, right, who sold chocolate, and the company is still here. A lot of this medical center was built with chocolate money, and there are chocolate kisses on the light posts all throughout town. It's really quite remarkable. And last night I was at the Hershey Inn, is it, for dinner? Is it the Hershey Inn?
Hershey Hotel.
Hershey Hotel. I've never been there. Remarkable place, especially inside. Pretty cool.
Beautiful. So
let's start with some histories of our guests today. Let's start with you, Leslie. I've known you for quite a while, and you're a retrovirologist, but I don't know where you're actually from originally.
Well, I grew up in western Pennsylvania, a small town called Charleroi. It's south of Pittsburgh.
Okay. And where did you go? You went to high school locally, I guess.
I went to high school there.
Where'd you go to college?
I went to Dartmouth College.
And were you a science major?
I was a psychology major with modification in biology. I was interested in biological basis of behavior.
At the time you went, so you're an MD, so were you thinking already of going to medical school when you went to college?
I went with an open mind. I mean, I come from a family with lots of physicians, so my grandfather, my father, my brothers. So I'm sure it was in my mind to do that, but I kind of thought I should explore some different things because during college, you have that opportunity.
Yeah,
you can study so many things. And then I guess I did an honors thesis. And to do that, you had to do a research project. So I chose to do that in psychology department. And it was on learning and behavior. And I really enjoyed it. But what really was so interesting to me is when I went to the seminar at the end of the year where all the other students showed their work, there were a lot of people starting to work in molecular biology, and I was absolutely fascinated by that topic. So that was, I think, the first time that I thought, I am so interested in this field, I could really do this for a career. But I didn't really have enough experience to go as an MD PhD, so I went to medical school.
Where'd you go to medical school?
I went to Duke, and the reason I chose that was because there was a year offered for research. So it's a curriculum that's some schools have adopted now, but we had an entire year off. So during that year, my summer between M1 and M2, I actually went to the NIH and did some research there. And so when it was time to do my entire year, I actually applied. There was a Howard Hughes program that was at the NIH during that time. So I applied to that program.
So in your years in medical school, were you thinking of being a clinician or doing research?
I was really thinking of trying to do both.
Both, yeah.
Yeah. After the experience at the NIH where I worked with Dina Singer and really did molecular biology in the immunology branch, I fell in love with research. And I thought, I really need to give this a try.
So after medical school, what did you do?
Well, after medical school, I went back to medical school. And, I went to medical school in 1983, which, that's when I started. That's the year that the HIV virus was discovered. And I got very interested in it clinically, but then also in research. So I stayed at Duke for my residency, and I did an abbreviated clinical track, and then I did 2 years of research. And I did that with Warner Green, and we were working on IL-2 receptors and HTLV and retroviruses, and that's when I really thought I wanted to try to make a career of it.
Okay. So after that, where did you go next?
After that, I came here, and I did an infectious diseases fellowship. And then I did a postdoc with John Wills. And I have to say that the entire Department of Microbiology and Immunology really welcomed me, and I think that was the turning point for me. They were incredibly supportive. They thought it was really interesting that an MD would come and try to do research, but they found that it was something that intrigued them and that they really wanted to see it be successful. And there were a cadre of virologists here that were just wonderful mentors for me. Even, people completely outside my field who just took a great interest in helping me to learn more about the scientific process, grant writing, paper writing, giving seminars. They really took me under their wing, I would say.
And then after your years with John Wills, you became a faculty member here.
I became a faculty member, in the Department of Medicine, and then I had kept my joint appointment in microbiology, immunology.
So you've had a lab ever since, and we're going to talk about some of your work on retroviruses. But now you're also a dean here, right? What kind of dean are you? Can you tell us your title? Yeah,
so 3 years ago, I became vice dean for research and graduate studies. So in that role, I oversee all of research. I have a huge staff of tremendously talented people who really do all the work, and I kind of just answer hard questions when they come up. And then I also oversee the graduate school, but we also have an associate dean for graduate studies who, again, does a lot of that.
And you continue to have a lab, right?
Yes. So I guess I'm around year 20 of my grant. I'm hopefully going to be renewing that soon. So yes, I've had a lab ever since I left my postdoc. I've had an active lab.
So do you want to be a dean as long as you can, or would you like to someday just go back to having just a lab?
No, that's a great question because, I have such a passion for the research. I love it. It's so exciting every day. There's, new things happening all the time. In my vice dean role, I actually really love that too because I get to not just accelerate my research, but I really get to amplify everybody else's research. So, the ability to do that and the fun programs that we can develop that really fosters the development of, junior people like Nick, but also making sure that senior investigators have the types of core facilities that they need and infrastructure. Trying to put people together, interdisciplinary research, I think is really important. So we're building a lot of programs to try to get collaborative research going in different fields. So that's really exciting and that's fun, and that is, certainly beyond me. And I think that's why I like it so much.
And I should add that she's doing a great job. As a faculty member, we feel like she has our best interests at hand.
That's great. Well, nothing like one of your own to do this kind of position, as opposed to someone who's been in administration their whole career and has no sense for research, right?
Somebody who is interested in virology really helps too.
For sure. I can't think of someone in a similar position at Columbia, so you're very lucky. That's good. Now, in all these years, research and research plus dean, have you also seen patients?
Yes, actually I was on service last week, so.
And you're in infectious diseases? Infectious
disease. I just do hospital consults. I have done outpatient in the past, but now I focus just on inpatient consults, and that is actually a sort of, I think about it as a little bit of instant gratification versus research being a little bit more delayed gratification, so I kind of get to feed both halves of that.
Sure.
And plus I work with the fellows, the students, the residents, as a teaching hospital, it's really a lot of fun to work with the trainees and the students. That's the best part of it.
So I always like to ask MDs, if you're on an airplane and they ask for a doctor, will you get up?
If no one else does. But usually there's somebody else who's an ER physician or, someone who feels a lot more comfortable. But yes, I definitely would if nobody else with without more skill.
I asked someone that. I asked someone that, and I can't remember who it was, and they said they would hide.
I mean, if I'm with my husband, I tell him to go, because he's also a physician.
An MD.
But yes,
it depends on how much you've. There are many MDs who simply do research and have no more clinical duties, and so they don't feel comfortable doing that. So, okay, Nick, where are you from?
I'm actually from Northeast Ohio, so a suburb of Cleveland area. Mm-hmm. That's where I grew up. Went to school there.
Did you have science influence in your family? Leslie said you had a doctor as a father, right?
Right.
Did you have any science influence?
I really didn't have science, but I guess you can say my aunt was a PhD. She trained with some great scientists, Ed Harlow and Carol Grader. So
cool.
So I had peripheral, but really my dad's in computers and my mom's she's a nurse. So I guess science there a little bit.
Computer science, yeah. My kids always tell me, it's a science, Dad. Where did you go to college?
I went to college at Brigham Young University.
Okay.
Yeah.
Provo, Utah.
Provo, Utah.
Right. And were you a science major?
I was a molecular biology major, actually. I didn't start a molecular biology major, but I did some internships during summers.
Right.
And that's when I fell in love with research. So I went back and changed my major to molecular biology.
Ah. So what were you thinking of doing? You weren't thinking of medical school, right?
Maybe it was it rotated through. I didn't know what I wanted to do, really.
Yeah.
I hadn't found my passion until a couple internships. And then I knew research was what I wanted to do.
So throughout college, you said, I want to do research and eventually apply to PhD programs, right? So where did you go for your PhD?
University of Pennsylvania. So I went, applied to their graduate program. And again, didn't know what I wanted to do. I knew I wanted to do research, but you read all the faculty profiles and you say, oh, that's really cool. I could do that. And you read the next one, and you're like, oh, that's really cool. I could do that. And so of the 200 plus professors, I hadn't narrowed it down at all. You can't do 200 rotations.
Yeah.
But I'm very happy with how things fell out. And I met Jim Allwine and did my work with him. And I think that worked out well.
I was going to say, I know that you worked for Jim, and I've known Jim for years. And he's a great mentor.
Oh, yeah.
The reason I remember Jim is because at Gordon conferences, he would have posters. His poster, he'd stand before a poster, even up till a few years ago. And he had a bottle of sherry and cups. So whoever came by, he would offer them and they would stay.
Sounds like the Jim I know. So he has a tradition where when you graduate from his lab, you give him a bottle of scotch. And then when the next student graduates, they open up the old scotch and he gets another bottle. And I guess I have the distinction of giving him the last bottle of scotch. I'm his last PhD student to graduate, and so we opened it as a retirement party.
Nice. Because you're retired now.
He did, yeah.
So there you worked on cytomegalovirus,
I did,
right?
cytomegalovirus, yeah.
Okay. And where did you do a postdoc?
I then went to the lab of Scott Emmer. Now, Scott is not a virologist. He's a yeast geneticist. Maybe I shouldn't admit this on TWiV and a virology podcast, but when I was done with graduate school, I was ready to be done with viruses. But they're infectious, and I want to away from them. It didn't take long to realize that I missed them dearly, and so then I started wanting to, as I was a postdoc, thinking about how I could get back into viruses. But it wasn't too much of a stretch because I was working on the Escort machinery. And we know that there's a big link to viruses there. Yeah,
and we'll talk about that. And then after your postdoc, you came here.
I did.
How long have you been here now?
Over 4 years.
Okay.
I came in summer of 2000.
So I want to ask you, so you did a postdoc in yeast, essentially, right? And then when you applied for jobs, did you say, I'm going to work on viruses?
Yeah.
And how did that go? Obviously, you got a job here, but
it must have worked.
In general, was it well received, or did people say, "Oh, you have to work on Escort only"?
Yeah, I think it was well received. I think it was combining two related fields and maybe having a different. perspective, and there's an advantage to that. I wasn't going to compete with Scott because Scott's not a virologist. I think, that was big because he would destroy me, being the big, powerful, great scientist that he is. So having my own niche and being able to apply some of the techniques that I learned well in that lab, because even though it was a yeast genetics lab, I learned a lot of biochemistry techniques that, in fact, if you looked at my papers there, they're probably a lot more biochemistry than yeast genetics. And so that's been helpful. And we have biochemistry projects in the lab now and trying to figure out, using the same techniques that I used as a postdoc.
Yeah.
And, I always joke, I only needed to convince one department that, it would be a good fit.
Of course. Yeah. No, but as we'll see, it makes perfect sense because the virus you had worked on as a PhD requires the Escort pathway. So it was a perfect thing to put together, and so I'm glad it worked. So we'll talk about that. Now, when you in Scott's lab, you were in Cornell in Ithaca, is that correct?
Yes, we were in Ithaca.
Because I know he had previously been in California.
He did, and they started a new cell biology institute there that Scott went to become the director of. I think he's having a lot of fun doing that. So we were in Ithaca.
All right, so Leslie, let's talk about your science. Now, I know you because of a retroviral protein called GAG, and in fact, a couple of years ago, I invited you to ASV to be a plenary speaker because I knew of your work on GAG. So let's talk about that a little bit. So whenever I prepare for TWiVs, I go on to PubMed, which is a great resource. I don't know what we did without it. We used to go to the library. Oh, you used to get these booklets in the mail, which were searches of the literature, right? You remember those? And you could look through those, but you just had to look through journals, and now it's so much easier. So PubMed, I looked for your papers, and the first virus paper I could find, and you could correct me if I'm wrong, because I often get this wrong, because I'm not great at PubMed searches, is a 1994 paper with John Wills, and it's identification of a membrane-binding domain within the amino-terminal region of human HIV type 1 GAG protein, which interacts with acidic phospholipids.
Yeah.
Is that your first virus paper?
If you say so. I didn't look at my own. I didn't PubMed myself. I trust that it probably is.
So here we have, let's explain what GAG is to people who are listening. And this is HIV GAG, but all retroviruses have GAG. What is it?
Yes, they do. So GAG is the main structural protein of the virus. But the part of its function that we're very interested in is how it orchestrates the process of building a new virus particle. So it's a protein that is made in the cytoplasm, and we can talk more about what we learned later, but it goes to the plasma membrane, it recruits the RNA of the virus for packaging, it uses the escorts to get there, and then it buds from the plasma membrane. So it has a very interesting kind of life cycle within the cell trafficking cycle that it does, but it's absolutely required for making a virus particle. It is what does all the work, if you will.
So I guess you mentioned earlier you went to John Will's lab to do a postdoc, and that's where you got interested in GAG because they were working on it.
They were, yeah. John had done some really seminal experiments looking at different domains within GAG and trying to understand what the different regions were involved in. And you can just express GAG by itself, and you can get virus-like particles. So it simplifies the system, which is really important to do. So he and people in his lab were able to identify the region of the protein that's important for membrane binding, which this paper talks about. There's a region that's really important for GAG-GAG interactions, so protein-protein interactions, and a region interested in protein-RNA interactions as well. And then a region that I was somewhat involved in later that was the late domain, which is the late pinching off step where the escorts come into play.
Escort involvement, right. So GAG stands for what?
Group-associated antigen.
Antigen, yeah. So that's
it's not the best name, but
it was
what we have
given
to work
years
with,
ago,
so,
right?
Yeah,
Before we knew anything about
because if you look at antibodies against these viruses, that's the major antigen that is detected.
Right.
So if we look for, for example, HIV infection, the ELISA has the GAG antibodies that we're looking for in addition to some others.
Right. And GAG, you mentioned, has multiple domains, right?
Yes.
What are those?
Well, it has the matrix or the MA domain, and then there are some smaller proteins that can be there, and then capsid domain, which is CA, and nucleocapsid, which is NC, and then HIV has this extra P6 domain.
Right.
So they have those three that are conserved, and then they might have some smaller peptides in between.
So in this paper, why is it important to find a membrane binding domain in the amino terminus of GAG? What does that tell you?
So in this paper, we were looking at what was required for HIV GAG to go to the plasma membrane. Because people, and to be totally honest with you, it hasn't been completely worked out, every single step of that. But there is a very strong propensity for this protein to traffic to the plasma membrane. How does it do that is really the question. So it's a cell biology question, but it's also using a virus to answer that. So we did work with a scientist, Marilyn Resch, and her group, who had looked at the SARC protein and looked at how it was able to get to the plasma membrane. So it uses both the basic residues that are within the N-terminus of the protein, and then also myristic acid addition.
Right.
And so that bipartite signal is what's important for driving these proteins to the plasma membrane. So
HIV also has myristate? And
it does, and if you knock that off, then it doesn't go to the membrane. So it was sort of thought of as a, a potential way to disrupt budding.
Right. So I teach this, and I often get the question, well, even though GAG has this N-terminal signal, it's just floating around the cytoplasm. Is it just random that it gets up to the plasma membrane? Is that how it works? Or is there some directed transport?
Yeah, so there's a lot of work that's been done on things like, microtubules, actin, interactions with Escort machinery, and certainly all of those probably play an important role. I don't think it's random. It does seem to be a very directed process, but it involves many of the cellular components that we talked about, and RNA binding is important as well.
So if you disrupt microtubules, you will disrupt the movement to the plasma membrane of GAG?
Yeah, I've not done those experiments, but...
That's others, yeah, okay. And you mentioned that GAG, if you produce just GAG in cells, you can make particles.
Right.
So you make the whole precursor, and that goes to the plasma membrane because of this hydrophobic signal, and there's no RNA, so you make empty particles.
Well, there is RNA. It's just not viral RNA.
It's not viral, so other RNAs will get in there.
Yes, a lot of cellular RNAs get into the particles.
Oh, even without a packaging
That's
signal.
right. Yep.
And then this protein drives the budding along with Escort, as we'll talk about. Yes. And then and that's the basis for making vectors, of course, right?
Absolutely.
With different viruses. So it's interesting that without a packaging signal, you get other RNAs in there. It's just because they're captured randomly, right?
Well, there's been a lot of recent work.
Some people think
some older work kind of set that up as, there's a lot of cellular RNA in the particle. Yes. But there are some that are preferentially packaged. There's some small RNAs that are packaged. Alice has worked on.
Okay. Yes.
And then Paul Binasha's lab has recently done some really elegant studies with a PAR clip showing basically all the RNAs that are there, and tRNAs actually interact with this matrix domain.
Right.
And so probably RNA binding in general, there's both nonspecific and specific RNA binding properties of GAG. And so the nonspecific or lower affinity, maybe is a better way to say it, recruits a lot of these RNAs. And then the specific signal in NC recruits the genome itself, the RNA genome.
So if you're using lentiviral vectors, and you put a gene in via having a packaging sequence, are you also going to get cellular RNAs?
There'll be some cellular RNAs.
But for gene therapy or whatever the application, that's not an issue, right?
It doesn't seem to interfere.
Yeah.
Now, do they play a positive role was, I think, a harder question to ask, but it doesn't seem to interfere. So
looking through the rest of your papers, you spent pretty much the ensuing years working on GAG in some way or another. Is that fair to say?
Yeah, yesterday I said we haven't strayed very far from that. Yeah, we've continued to follow it.
I think a really important paper, and the one that I remember you for and why I brought you to ASV, was this 2002 paper where you show that the GAG actually goes in the nucleus, which was surprising, right? So tell us a little bit about that.
Yeah, it was surprising. So that was studying Retroviruses virus gag protein. So, I did early work in HIV, and then mostly because of this study, I kind of turned more to focus on RSV. And so, yes, so we were very interested in this trafficking question that we talked about before. How does a protein go to the membrane? And so we just did a simple thing. We just took different segments of gag and put GFP on it and asked, where does it go in the cell? And MA tended to be in the nucleus and in the cytoplasm, but when we added this other piece on, which is just called P10, it has no real name other than just protein of 10 kilodaltons. Suddenly the protein was outside the nucleus. And so we didn't know if it was just too big to get in or if something was sending it out. And so my student at the time, Lisa Scheifele, she did experiments where she looked at bigger and bigger pieces, and we were pretty convinced that it probably was going out. So at the retrovirus meeting, a colleague suggested that we try this new drug, leptomycin B, to see what would happen. So she did all the control experiments, and it was very clear that if you inhibit nuclear export, the gag protein just accumulates in the nucleus.
So leptomycin is an inhibitor of export, right?
Yes, inhibits the CRM1 export pathway. So that was just very surprising. I can remember the day. You remember these things because, you don't have sort of surprising discoveries every day. But, she was down at the microscope and came running up and said, you have to look at this. Look at these pictures. So that was a very fun day. We were very confused about what was going on.
So you're looking at GFP signal, and you saw it in the nucleus in the presence of the drug. And this is Rouse sarcoma virus. Does it happen with other retroviruses as well?
Yes, it does happen with other retroviruses as well. Not necessarily to the same degree, and not necessarily all of them are not leptomycin B sensitive, but yes, like feline immunodeficiency virus and others. And then the foamy virus, actually its gag is in the nucleus without the drug even. You can see it.
So to get in the nucleus requires an import signal, I presume?
Yes.
And then to get out, it needs an export.
That's right.
So if you alter those signals, say if you prevent nuclear entry, does that block virus replication?
So because there's so much overlap in these signals, because viruses, and this virus is not a huge genome, so it has many duplications and functions. So the membrane binding domain in MA is also the nuclear localization signal. And there's a second nuclear localization signal in NC, but it's also the RNA binding domain. So you really have a lot of limitations in what you can mutate without affecting another function. So what we did instead was we took a mutant that John's lab had made a long time ago that had the SARC membrane binding domain at the N-terminus. And so we redirected the protein away from the nucleus. I mean, it still goes in, but not to the same degree. And in that experiment, we could see that you then reduce RNA packaging. If you then add a new nuclear localization signal elsewhere in the protein, you can restore that packaging. So that said to us, maybe this has to do with RNA packaging. Since RNA is made in the nucleus, maybe the protein's going in and capturing it there.
Right. And that whereas the previous model had been it captures it in the cytoplasm.
That's right.
And so here you're going in the nucleus. So GAG is made in the cytoplasm. It goes in the nucleus. There it grabs viral RNA, which is transcribed, obviously, from the provirus in the nucleus, right? And then the whole thing gets exported.
That's what we're still working on. I know it's been a long time, but it hasn't been tremendously straightforward.
Yeah.
So yes, that's the best evidence that we have for the model, and we do have some new unpublished data that's highly suggestive of that.
So do you think any GAG RNA interactions also occur in the cytosol?
They certainly could, yes. We're not saying it's the only way, but we do see GAG RNA interactions in the nucleus, and if they're disrupted. But it's absolutely possible that they also occur in the cytoplasm and maybe even at the plasma membrane. So
this, I call this a nuclear experience.
Yeah.
You probably do too, right?
I do.
So some other aspects of eukaryotic biology, some translation proteins require a nuclear experience.
Yeah, nuclear
go
experience
in and out.
matters. Yeah.
That's very interesting. So your idea is that it's picking up. So do you, if it can happen in the cytosol, if the RNA combined, I guess it's NC, nuclear capsid in the cytosol, what's going on in the nucleus that's so essential? Because as you said, if you block that, you block infectivity.
Yeah, you block the packaging, right. Well, we've thought about this a lot. And the interesting thing about retroviruses is that their genome is unspliced. So they have to capture unspliced RNA. And so most of the unspliced RNA, of course, is going to be at the transcription site because it does get co-transcriptionally spliced into another form that is packaged but much, much lower level. So part of our idea is that if you want to find something, go to the source. So you want to go where the most of something is present and you know exactly where that is, that's at the transcription site. So that's our model, and that's how we explain why it would be preferential to do that, because once the RNA goes into the cytoplasm, it's very diffuse. It's just in all kinds of locations, and there's no real bright signals for that. So you would have to imagine a way that GAG finds the RNA in the cytoplasm, and maybe there is a specific location, but that hasn't really been identified. So if you want to know where the highest concentration of unspliced RNA is in the cell, it's at the transcription site.
So are there any retroviruses where GAG does not go in the nucleus?
Oh, I'm sure there are. There's, examples, many in the literature of those that do, but it's a small amount. Just like with RSV, if we don't use the drug or make a mutation, it's 10 to 20% at any one time. So it's a very small amount, and studying small amounts of things, as you know, is somewhat difficult. So if we do very careful imaging studies or fractionations of cells, we can see other GAG proteins in the nucleus, but not easy to study.
And the leptomycin experiment still implicates the nuclear experience as being important.
Well, for RSV, but as I said, most of the other ones don't have a strong effect of LMB.
I see. Okay.
So I'm just
wondering if you can learn something from those viruses that don't need a nuclear experience. Right?
Absolutely. Yes, we're trying to compare different viruses to learn about that.
So another paper, 2013, you implicate the nucleolus in trafficking. Is that correct? Nucleolar trafficking of MMTV GAG induced by interaction with a ribosomal protein. So is that correct, that you think in some cases the RNA is in the nucleolus?
Yeah, so you mentioned, comparing other viruses might be helpful. So primarily we've been looking at MMTV and HIV and trying to compare and contrast. MLV actually makes immature particles in the cytoplasm, so it has a slightly different trafficking pathway. And so a collaborator, Tatiana Galivkina, had found with Steve Goff a yeast-2 hybrid interaction with this protein.
That's right.
It was a ribosomal protein.
Goff is on this paper. Goff is my colleague.
Both of them are obviously involved in the paper because they found the original study, and then we worked with them to look in cells if we could see an effect. And so we did see that there was an effect. If you express this protein at higher levels, you could kind of saturate the interaction so that you could then see it in the nucleolus. Right. It was very hard, again, to really understand what the true biological relevance is of that interaction. So again, those are studies that are still ongoing. And we see other GAG proteins in nucleolus, HIV, RSV. Again, it's hard to know what the function might be. But again, it's at very low levels under a steady state condition.
Got it. So then there's another interesting paper I wanted to touch on. A murine retrovirus co-opts YB1, a translational regulator and stress granule associated protein, to facilitate virus assembly. So here you mentioned these yeast virus-like particles called TY1 and 2. Maybe you could tell us a little bit about those.
So Suzanne Sandmeyer, another colleague, has done just tremendous work with that retrotransposon and has really shown a lot of interaction between the GAG protein and all of these RNA binding proteins in the cytoplasm that are involved in stress granules and P bodies. It's really beautiful, elegant work. The genetics are fantastic.
So let me ask you, these are retrotransposons in yeast, which means they are elements moving around the genome via reverse transcriptase. They're thought to be the precursors of retroviruses in mammalian cells.
Yeah, they have a lot of similarities, some differences, but yes, a lot.
And they make particles, right?
They don't get released from the cell.
They make intracellular particles. It's curious, right? Why would they do that?
Yeah, it's probably involved in the whole retrotransposon process.
Okay.
Because they do, of course, jump around within the genome.
Okay.
So it's probably involved there.
So this paper talks about YB1 and for assembly. So YB1, you say, is a translational regulator, and it's involved in assembly of, is this MMTV or?
Yes, with MMTV, those were those studies. So yeah, YB1 has a lot of roles in the cell. Actually, it's one of those kind of multifunctional proteins. And we did find that it interacted with GAG in the cytoplasm at distinct sites, and our hypothesis is that those are. I mentioned that MMTV makes immature particles in the cytoplasm. Right, those are associated with those sites, and potentially has a helpful trafficking role in getting out.
But this YB1 doesn't have a role in the yeast transposon in any way, right?
I guess I would have to look a little harder if anything's been done since then, but there were some pull downs that were done, but I'm not sure about the functional studies.
So how do you think YB1 works in virus? Like, is it a chaperone of some kind? Do you know?
Yeah, that's what we wondered. I mean, we wondered if, because it has a strong RNA binding element as well, we wondered whether it was a role in kind of helping to sequester the viral RNA in a compartment where GAG can find it, because as I mentioned, it's hard to understand how they find each other. So we wondered whether YB1 was helping to have the untranslated, because the unspliced that's out there in the cytoplasm, there's a translating pool and a non-translating pool.
Oh, okay.
And so maybe this is helping, like in stress granules where you are having translational repression. Maybe that was part of it, is that you could take and remove the RNA from the translating ribosomes into another compartment that then GAG would traffic there, and they could meet up in that location.
Right. Got it. Okay. So you have also been interested in RNA packaging.
Yes, right? More so today than before, yes. And
so tell us, what's the problem? How does that work, first of all, for retroviruses?
Well, it goes back to what we were talking about previously about localization. But basically, the untranslated RNA needs to be packaged. And in HIV, it has an accessory protein called Rev that helps to get that RNA that's unspliced out of the nucleus. Because normally you don't want an unspliced RNA to leave. And so that's one way to get the RNA out, which could be used then for both translation and packaging. And again, like, how does that split happen? Is one marked differently than the other? Are, I think, still some open questions about that. So that packaging that occurs then has to be a way for GAG to specifically recruit. And the interesting thing, too, is that in a virus particle, there's only 2 copies of RNA. But there's thousands of copies of GAG. So obviously not every GAG is binding an RNA in the cell. There's a minority of GAGs. So a small amount of GAG binding the RNA in a location does make sense for the biology of the virus.
So I remember that the Maloney murine leukemia virus packaging signal is pretty simple. It's a single size sequence.
Yeah, the size sequence is present in almost, I think, every retrovirus.
And that was discovered in the Baltimore lab when I was a postdoc there. One of the students actually discovered that. But it's more complicated for HIV, right?
Yeah, there are some long-range effects that seem to be important as well. So, there is always the core packaging sequence, but some of these downstream RNA elements are probably important. I mean, sometimes, we have to remind ourselves that RNA is not just a long string. It has a very complex, 3-dimensional structure. And so some of these downstream interactions certainly are important for packaging. They may not have quite as big an effect in isolation, but they all are additive.
And these size sequences, PSI, the Greek letter, right? Which I guess the P for packaging,
I think so, yes. It was called E at one point for encapsulation, but I think Psi took over after that.
So they bind nucleocapsid, that's the idea? And in your case, some of that happens in the nucleus.
Well, that's our proposal.
That's what you're thinking,
I wouldn't say we've nailed that down yet, but we're working on that.
Yesterday you told me you have some exciting things in press.
Well, not quite impressed, but ready for submission.
Do you want to talk a little bit about that?
Yeah, I think I was mentioning to you that, I kind of believe that you go where the science leads you. And so that's what's really fun about a career in science, because you don't know where it's taking you next. So we didn't plan to go into the nucleus. That wasn't our goal. So I had to learn a lot about there's so much new about the nucleus. It's absolutely fascinating. But we were really interested in sort of the way that the nucleus compartmentalizes substructures within the nucleus. So like splicing speckles and paraspeckles and some of these other nuclear bodies, they don't have membranes. So it's really been very interesting that people have found that most of them bind an RNA, and they form these liquid phase separation or liquid-like droplets that allow them to coalesce into very specific sites and to freely exchange, but they don't have a membrane. So it's a relatively new area of nuclear biology that I think is really fascinating and kind of can explain maybe how the nucleus functions as it does. Again, it's not just a big, sack of DNA with some proteins thrown in. There's a lot of organization that's come out that's very fascinating. So we're trying to understand how that plays into what we're seeing with GAG in the nucleus and its interactions.
I read recently where, I don't know if you know this, but it's interesting to think about how the nucleus arose, right? Because the earliest cells didn't have the archaea and the bacteria didn't have, but the idea is that a bacteria invaded an archaeon many millions of years ago, right? And became mitochondria. But there were all the other bacterial genes, and they had to be hidden, and that's how the nucleus arose in response to that, which is kind of an interesting thought.
Fascinating.
This concept you mentioned where you let science lead you, or you follow where it leads. That's why I think it's a privilege to do science,
it is.
Right? Because where else can you just follow something apart from what, say, you're funded to do? And we can do that.
It's absolutely true.
If you were in a company and you started doing something else not related to your job, hey, stop that. So that's why I say being a scientist is a privilege.
It is.
Right?
It is a privilege.
It really is.
And one of my initial first mentors told me that, and it stuck with me so much. It's an absolute privilege. Yeah. We're very fortunate.
So let's talk a little bit about Nick's work. I think we should start with your PhD because you're back to human cytomegalovirus, right? And I found a couple of papers that I want to chat with you about. So you went to the Alwine lab. They're working on HCMV,
yes.
Right? So that's why you work.
Yeah.
That's what we all do. We work on the things that the labs work on when we go, we go into them. So here's a paper from 10 years ago. I don't know if you'll remember, but it's not that long. Cytomegalovirus specifically controls levels of the ER chaperone BIP, which is required for virion assembly. So that caught my eye because BIP was the first protein shown to be translated in cells infected with poliovirus. Now, do you know why that is? I can tell you if you don't know.
Go ahead.
Polio inhibits cap-dependent translation.
And BIP has an iris.
And BIP has an iris. Yeah. So Peter Sarno discovered that. He actually took polio-infected cells, pulled out the polysomes, and then did microarrays. Back then, we couldn't do RNA-seq, so you had to do microarrays. And he found BIP was a major mRNA. We were all surprised because we didn't think there were cellular irises, internal ribosome entry sites. Yeah. So tell us how CMV regulates BIP.
Right. So right before I joined Jim's lab, they had a paper showing that CMV activated the unfolded protein response. So one of Jim's interests is, this virus takes about a week in cells to replicate. And how does the virus keep the cell happy so long? It's still in all of its nucleic acids and its amino acids and stuff. How does it keep it happy? So he was looking at stress responses. And the one that gets activated in the endoplasmic reticulum, the unfolded protein response, BIP is a major player in that and gets upregulated as part of that. But you have to keep in mind some of the results of the UPR signaling, for example, a stop in translation would not be good for the virus because it needs to translate. It uses the cellular apparatus for translating its proteins. So Jim wanted to look at the different parts, how does the virus turn some things on and other things off so that it can kind of optimize the conditions for its replication. And so when I joined the lab, he went through some of the data. There had been a paper maybe a year or two before I joined, and he said, see what you can do with this. And so we were looking
That's typical, right? See what you can do. Goodbye.
Here's your rotation. See what you can do, and if you do a good job, maybe you can come back. But anyway, so we looked, and BIP clearly went up a lot. And so we said, okay, well, the next thing is let's get rid of BIP and see if it actually adversely affects the virus replication. And so we did that, and we did that in a strange way. So there had been a recent paper about a bacterial toxin that had come out of Australia, and it actually cleaved BIP, and it made it non-functional. And that's one of the reasons we get sick when we get infected with E. coli and stuff. And so we called and had them send us some, so we could just simply add it to the cells, and we could show very nicely on a Western blot that the full-length BIP disappeared, and we could see these fragments, and it was no longer functional. And when we did that, we completely stopped the virus replication. And so we were excited about that. And then we did it other ways with short hairpin RNAs and things, just to show that it wasn't some other thing that the toxin was doing. And so we knew BIP was important. And we started to look at what was going on. And we could see in the nucleus, you had all kinds of nucleocapsids formed. So we knew it wasn't blocking like entry or even DNA replication and the packaging of nucleocapsids. So we turned our attention to the cytoplasm a little bit later in infection, what was going on. And so a little bit of background about cytomegalovirus is that it forms this assembly compartment. And it's not a compartment like some viruses that's completely a membrane encapsulated or whatever, but it just takes its organelles and it kind of moves them towards the MTOC, the microtubule organizing center. And so if you look at an infected cell, it no longer resembles the organelles that you would see in a textbook. You've got this concentration of endosomes right at the center. The Golgi stacks kind of form a ring around these endosomes, and it's really cool looking by microscopy. So we started noticing that when we got rid of BIP, you just don't have that structure anymore. If you look at an electron micrograph, you can see lots of dense bodies, viral particles. There's just a lot of activity in that area that denotes that this is the assembly compartment. You can pick it out very easily. But the cytoplasm of a BIP knockdown or a BIP toxin treated that cleaved BIP was very quiet. It looked like an uninfected cell. But you could look in the nucleus and see all kinds of nucleocapsids. So we were really intrigued by that. And so we were able to show that you need a intact functional BIP in order to form this complex. And without that concentration of organelles and all that viral activity, you really make virus quite less efficiently. You need that for efficient virus replication.
The precise mechanism you don't know yet, I guess, right?
There's a lot of people that would love to know that, yeah. So
is that something you work on now, or?
We do. So there's been some great work from other labs, Phil Pellet's lab, for one at Wayne State University, that have identified some viral proteins that are important. Other labs have identified cellular proteins. So one of the papers from my lab, we identified a snare that was important for kind of building that process. So we're still in the identification phase of what factors are needed, and to get a mechanism would be fantastic, and exactly how the virus is doing that. And we and many other labs are interested in that.
You have another paper where you show that the way BIP is upregulated in infected cells is via a protein called LA.
Yes, and the iris, right?
Yeah, so that's also because in the early days of viruses, which were first found in picornas, I think it was Sonnenberg who found that LA, which is a component of ribonucleoproteins in the cell, right, is needed for some irises to work. So you find that in HCMV infected cells, LA goes up, right?
So we find that. So we were interested in BIP levels were going way up, and so we were interested how is this? Is it protein stability? Is it the microRNA level? And it is. The microRNA levels go up too, but we thought there was something more because it didn't seem to match quite the induction. There's really a ton of BIP coming up, and so we looked at if there was increased translation from this iris, and we were able to see that there was, and it was dependent upon the LA.
So LA goes up, right?
Yes.
Do you know how the virus upregulates that?
We did not pay attention to that.
Yeah. I would guess that other irises go up as well in the cell, because probably LA is required for more than just BIP, right?
Presumably, yeah.
Yeah. I wonder what other ones.
We were centric on BIP because of our interest.
Sure.
I'm sure, yeah.
BIP, by the way, is also needed, is a chaperone for polio assembly. And it may be that having an iris is why it can be used, right? Who knows what came first, but because it continues to be translated. But there are also other mRNAs that continue to be translated as well. And as you said, you have this other paper which says that BIP influences the assembly compartment, which you told us about. If you take BIP away via the drug or small hairpin RNAs, You don't get the
You
assembly.
don't get the assembly compartment. You get almost no activity in the cytoplasm. It's quite striking.
It's interesting.
We were surprised.
Yeah, that's a cool thing to figure out. All right. So, but then at that point, you're tired of viruses. So you went to Scott Emmer's lab.
CMV just takes too long to grow. You do an infection, you know, you have a couple kids, maybe some grandkids, and you come back, you harvest the infection, you know.
Well, I don't blame you. I mean, polio starts to make viruses in 2 hours after infection. How long does a retrovirus take?
Well, it integrates within about 12 to 16 hours.
Okay.
So then it makes particles after that. So it's pretty quick.
So we were studying these late events in the infection. If we studied entry, who cares, right?
Yes, that's right.
A couple hours
That's
to get your results. But.
Right. So tell us, let's talk about Escort a bit. What does Escort stand for?
Endosomal sorting complex required for transport.
Who made that name up? Did Scott?
So it came from Scott's lab, and he had a competition, $50 for whoever came up with the best name. And they all voted on it, and I guess came up with different names, and they voted on him. He said it. He told me once it was the best $50 he ever spent.
So he actually paid it.
Oh, he paid it.
That's
as far
good.
as I know, he paid. I wasn't in the lab at the time, but as far as rumor goes, he paid it. Yeah.
So is he a key player in discovery of the Escort?
Yes. So he Scott's done a lot of great screens using yeast libraries, and he comes up with great ways to get readouts on whether pathways are blocked or functioning when you do certain things. And so he did one that was called the vacuolar protein sorting screen, the VPS screen, and he looked for components that blocked the delivery of a cargo to the vacuole, which is like the human lysosome where things go to get degraded and die. And he came up with a lot of screens. Some of those were expected, and then there were some that I should say he divided them into different classes. So class A, class B, and so on and so forth. And what's called the class E, and they're called that because of the phenotype. You instead of getting your cargo in the actual inside of the vacuole, they kind of form like this ring and this dot. So they're kind of trapped and accumulated. And so the class E mutants then are what make up the Escort machinery. And so there are 5 different Escort complexes, Escort 0, 1, 2, and 3, and then the VPS4 complex. So yeah, so Escort 0 came around later,
You had to go back.
Had to go back to 0. Fortunately, as far as I know, they're not in the negatives yet, so they haven't found any more precede 0. But and so each of these complexes, they have different functions, and just like anything in science, the more we learn, the more complex it gets. But from a simplistic view, Escort 0 and 1 have ubiquitin binding domains. And so when you're thinking about delivering a protein to the lysosome, which is just one of the many functions of the escorts, when you think about it in that context, they can kind of concentrate the cargo, kind of select the cargo, give it maybe some specificity for what's going to get degraded. And then at the endosome, you have Escort 2, which also has very little ubiquitin binding domains compared to the other ones, but it kind of comes involved. And then you recruit Escort 3 and VPS4. Those are the final 2 complexes. And that's really my interest was in. Then what I studied as a postdoc was the Escort 3 complex. And those are the ones that I think kind of get this to the point where scission occurs or is involved in scission. Different models. We still don't know exactly how scission results from the Escort machinery. And there's just recently a paper that says the Escorts actually leave before scission occurs. So maybe it just gets the membrane close enough, and it can either abort or cause scission. And then if it aborts, they have to come back and do it again until they get to the point where you're close enough to actually scission those.
So basically, you have 2 vesicles in a cell. The Escorts help bring them together.
Well, really, you have think of it as one membrane in the cell,
right?
And you're going to make a little vesicle inside.
Okay, got it.
So if you take your finger and kind of poke it into a balloon, that's kind of what the Escorts are doing. They're kind of you're wanting the cargo to go into the inside the balloon, and then you have to cut it so you actually have a piece of the balloon inside the balloon.
Got it.
And so the Escorts are what particularly the late Escorts are what are involved in that scission step.
And when you say Escort 0, 1, 2, 3, these are multi-protein complexes.
Multi-protein complexes, and the yeast, the mammalian have more isoforms than the yeast. So as you go higher up in complexity in organisms, you get more and more isoforms and more complexity, which makes things more difficult to study. But that's what they do. But since they were originally discovered in sorting things to the lysosome for degradation, then They've been linked to viruses now because it's the same type of thing a virus wants to do when it gets its membrane envelope. Cytokinesis, it kind of closes that final excision at the neck. In nuclear membrane repair, so if you get a hole in your nucleus, the escorts can come in and kind of repair that. And they're popping up all over as really useful tools to have in the cell to keep things in homeostasis.
So Scott discovered this whole pathway in yeast, right?
Yes.
And who found it in mammalian cells?
Well, I think different labs then would find the homologs. I think the major well, maybe I'm biased as a virologist, but the major, I think, key finding with the escorts in mammalian cells was that showing that it was involved in budding of retroviruses.
Mm-hmm.
Right.
And that came from Wes Sundquist's lab in 2001.
Carol
was a cell
Carter.
paper. Yeah.
You told me a story before about Wes. What was that?
Yeah. So I didn't know this when I was applying for jobs, but I came to interview in this department and spoke with John Wills, who's now a herpes virologist, but spent a time with retroviruses and late domains, and he actually coined the term late domain with GAG and the retroviruses. And he was telling me that they went and saw Scott, his wife, I think actually, Becky Craven, who's also a faculty member in our department, saw Scott give a talk and got thinking and said, I bet maybe retroviruses use that same machinery. And so if you read a PNAS paper from John's lab, there's a paragraph in there that maybe suggests that. And I didn't even know that was in there until I talked to John and he pointed out, and I went and looked up. Of course, it's in there suggesting maybe they use the same machinery. And of course, when there's a story goes that before Wes published his paper, he sent John an email that said, you were right. I haven't seen that email, but that's how John tells it.
So one of Leslie's papers, maybe this is it, that I thought we would tie in, positionally independent and exchangeable late budding functions of Rouse sarcoma virus and HIV GAG proteins.
Yeah.
So that predated the understanding that Escort was involved. But you have this late domain which had a late phenotype.
That was a very fun project. It was one of those, again, kind of those eureka things where, we were sitting in the retrovirus meeting at Cold Spring Harbor, and there was a presentation showing the kind of lollipop appearance of viruses that were stuck and couldn't bud. And we said, we got to go rush home and do these experiments, because we kind of got this idea that we had an idea that maybe this domain was involved in that. So basically we split up the project among a couple people in the lab, and we just hurried up and got it done. And it was really nice because this little motif that's in P6 and HIV, and that was the story that we were looking at, was also present in RSV. It was a different sequence, but these motifs could then be swapped for each other, which was pretty striking because they're very small. So that's where the positionally independent and exchangeable comes in. So that was really, that was something that we were kind of on the verge of understanding. And like you said, you just talk to somebody or see something and it pushes you over the edge, and you suddenly make a connection that you couldn't have made without that.
And of course, since these late domains are found in many budding viruses of all different sorts.
Yeah, all through
so they help
envelope viruses
come off the surface. So when cells make exosomes, are escorts also involved in that?
Certainly. There are escort-dependent and independent pathways.
Okay, so viruses have not invented a new way of getting out of this. So they've just co-opted something that already exists.
Certainly.
Okay. So in Scott's lab, you said you worked on late Escort 3?
Escort 3. We were interested in understanding the mechanism of scission. How was Escort 3 promoting scission? And so it was a very heavy biochemical project. I worked with another postdoc, Mike Kenny, who's now faculty down at UT Southwestern in the cell biology department there. We worked together to try to figure out if we could understand this mechanism. And so we had some good luck. We were able to visualize these filaments on electron micrograph and when we added the upstream escorts, it kind of changed their morphology. And it kind of made it like a ring. And then if you added more of the later escorts, it kind of gave it like a three-dimensional, it really looked like a slinky type like thing. And so we started to come up with models on how maybe that was kind of like pushing into the membrane. And there's always the question, VPS4 is it's a AAA ATPase, so it's the only energy input into the system. And it was known that that helped take the escorts off the membrane. But we didn't know if that was part of the scission process or, after the decision, or now even maybe with some of the stuff coming out before the decision, we didn't know. So we were really aiming to try to figure out this mechanism of this decision event. And we made one of the Escort that we focused on, Escort 3 proteins, is 240 amino acids. I think we probably made over 200 mutations of that protein. I mean, we know that protein pretty well inside and out. But through these analyses, we identified the membrane binding domain. We identified a helix that inserts into the membrane, which, when you insert a helix, you deform the membrane. Now, it was the opposite direction because, you have positive and negative curvature, and we're really thinking of a process of negative curvature, but maybe it's at the top. So we did, had some good luck looking at that. And I think a crystal structure that came out from a graduate student that we worked with who got the structure confirmed some of our membrane binding domain results and stuff. So it was fun. We really got to know this protein.
So in terms of methodology, you do some genetics, you make alterations in the DNA, you produce the proteins in cells, you must do some cell biology, fractionation and so forth, and then electron microscopy. Right?
Yeah, so a lot of imaging, how do these mutations affect its function, its localization, things like that. Okay. And then, like you said, fractionation, we showed it's on the membrane fraction, it's not on the membrane fraction with the different mutations. I mean, we thought we knew what the membrane binding domain was, so we changed. There was a couple lysines, 4 lysines in a row, well, not directly in a row, but on the same surface, using a model. And we changed them all to negative charges. It's going to blow it off the membrane. Right now, it stayed on the membrane. We were wrong, but we later found the correct membrane binding interface.
So as you're working in this postdoc, is the connection to viruses coming out in the press? Is that happening at this time, or had it already happened? Or
it had already happened, but I mean, you can always find new papers that say this virus requires it or this virus doesn't.
Yeah.
But the HIV work had already come out. The big paper that came out while working on this was that influenza does not use the escorts, that M6 does it by itself. So that was the big thing at this time that we're working on.
Had anyone looked at HCMV in escorts?
Yes.
They had, okay.
There was actually 2 papers. One was in 2007. So this is the time when I'm middle of my graduate work. I'm really interested in the rearranging of the assembly compartment. So I'm starting to think about trafficking. I'm starting to think about maybe doing a postdoc where I can do some cell biology and look at some trafficking factors. And there was a paper that came out from Mike Marsh's lab that said it's not involved. They used siRNA or shRNA to knock down Escort 3 and VPS4, and they never saw phenotype. And so then in 2009, while I was really getting ready to finish up in the lab, there's a paper that came out from Ed McCarsky's lab. Now, he's a big CMV virologist that said that if you block VPS4, it is involved. And so there was a little bit of, you never know, is it technical, is it, what's going on? We think, the paper from our lab, we think we've kind of understand what people were looking at now, and we're able to resolve that. So there was that work done on the escorts, and we knew that it was recruited the assembly compartment. I think a couple labs had shown that it was recruited there, so we figured it was getting there to do envelopment. But in a story that came later, we realized we were wrong with that.
So this is really what made you come back to combine Escort and HCMV, right? Because as you're working as a postdoc and learning that, you see that things are going on in HCMV, and you might as well go back to it, right?
Certainly. So
let's talk about 2 papers you published here. The first one is from 2016, potent inhibition of HCMV by modulation of snare syntaxin 5. Yeah. So what's that about?
So this one goes back, more relates to the work as a graduate student. When we were thinking about how this assembly compartment is formed, I got talking to my department chair, actually, who is a T-cell immunologist, uses mouse polyomavirus, and doesn't think about trafficking. I tease him all the time. He doesn't think about trafficking, but they learned about this inhibitor that blocks the entry of a lot of viruses, such as polyomaviruses, why he was interested in papillomavirus. It's been shown to block the entry of, and it was actually identified in a screen that blocked the entry of some toxins, some bacterial toxins, because they come in endocytosis, they get delivered all the way back into the ER. And so they identified this compound. And we knew that it probably wasn't going to block CMV entry, but I was curious as how blocking retrograde transport would affect the assembly compartment. Because we know that these organelles are moving down microtubules to the microtubule organizing center. And so we got some from him and put it on cells, and we were able to not completely inhibit virus production, but greatly reduce it. And so we looked at, electron micrographs. And so it was a little different than what we saw with the project I described earlier with BIP. With BIP, the entire cytoplasm was quiet, and you couldn't even tell there was a virus infection. This, you could tell there was components of a viral infection, but they were disordered, and there wasn't the formation of that nice assembly compartment. Like the pieces were there, but it wasn't getting put together. And so we went on to show that Syntaxin 5 was responsible for that. And we were interested in Syntaxin 5 particularly because when you undergo mitosis, your Golgi fragments, and then it gets put together after cytokinesis, it gets put back together. And there's 2 pathways that have been described in the literature that do that. And Syntaxin 5 is the only protein that's actually been described that's in both pathways. That's the same for both pathways. One is an NSF dependent, and one is a P97, but they both require Syntaxin 5. And so CMV is interesting in that it blocks the cell cycle, but it always has some expression of some mitotic factors. And so even though it's keeping the cell cycle in one state, it's expressing them from another part of the cycle. And we think maybe it uses some of the similar mitotic factors to break down the Golgi and then kind of reassemble it in this ring. And so there was a group, we had this hypothesis, and shortly after we started talking about it in the lab, of course, a group at UAB, Bill Britt's group, who's been studying CMV for decades, came out and actually showed that some of those Golgi structural proteins that get phosphorylated to break down the Golgi are CMV phosphorylates them to break down the Golgi. So, that was kind of validation of our hypothesis. Unfortunately, it wasn't validated by us, but Bill does good work, and so I think what the virus is doing is, again, not recreating the will, but just taking some factors that in a process that normally happens and using it to its advantage.
So Syntaxin 5 is not a part of the Escort pathway, right?
It is not. It's a snare protein.
Snare is involved in fusion.
Membrane fusion.
Right. What does snare stand for?
Soluble NSF receptor.
I don't know either. So.
I just taught this last week in a core class.
One of the students can tell us.
Yeah.
I'm going to look it up.
It's a soluble NSF-sensitive attachment receptor.
Soluble NSF attachment protein. Oh, that's SNAP. Soluble NSF, what did you say, attachment receptor?
Something like that. Yeah.
Okay, and it's involved in fusion of membranes in the cells, right? And a lot of that done by Jim Rothman, right? He got a Nobel Prize for that, I think.
He did,
right?
Yes, he shared it with Randy Sheckman, and then...
Right. The other paper you published, 2018, just this year, a non-envelopment role for Escort 3 during HCMV. What's that? So you expected escorts to be involved in the budding.
Absolutely.
Right? And this shows that it wasn't?
And I told my student, when he joined the lab, I said, all you got to do is block the escorts, or show that it's involved in envelopment. The paper that I mentioned in 2009 that said it was involved in replication, they didn't look specifically at envelopment. I said, we'll show its envelopment, we'll write the paper, you can graduate, life is good. Good thing he's not on the podcast. You probably have some choice things to say about that. No, he's a great student, and did wonderful work, and continually came back and showed, look, I expressed these dominant negative proteins, and either there was a phenotype, but that's because the cells were dying. If you can imagine blocking the escorts, the cells don't like that. Or there was no phenotype, and then, but it wasn't affecting the virus, and then I would say, you have to show me the pathway's blocked, and he'd go back, and he'd come back, and we just really weren't, we didn't know, we didn't get a phenotype over and over again, but we weren't sure that we were actually blocking the pathway. But the really experiment that he did in collaboration, again, with the herpes simplex lab down the hall, John Wills, who keeps coming up, was to show that even using his system to block the escorts, which was inducible, so we had control, timing of control, that you didn't get herpes simplex virus made, which had been published before, but you could get CMV made. So that was really the experiment that convinced us that we were blocking the escorts, but we could look at electron microscopy. We could see beautiful enveloped virions inside the cell. We could see it outside the cell, so it wasn't affecting egress. And, the titers were the same. And so we were, it was, he did a very good job of convincing me that it wasn't involved. But then we said, well, what's going on now? What is involved? And so there's two things that came out of this. One, we were thinking about the discrepancy in the literature. We agreed with the shRNA knockdown paper that came out in 2007. So it's like, okay, we agree with them, but what about the other paper? Well, we started looking at the techniques, and while the one did what we did, high MOI infection, look at infectious titers, the other one did a low MOI infection and looked at the spread throughout the monolayer. So I said, just for the heck of it, why don't you go do that? The same kind of experiment. And he did, and lo and behold, spread is actually much slower when you don't have the escorts. It catches up. So if you look 2 weeks out after you infect it, you get the same amount of virus, but in the middle of the time, it's very reduced. And you've already mentioned about exosomes. Escorts are involved in making exosomes. So he came up with the hypothesis that when a cell gets infected, it sends out messages to its neighboring cells via exosomes that says, I'm coming, get ready for infection. And the virus can go and infect those cells without those messages, but it just takes longer. And so he's got a really nice study that he's working on that will be wrapped up soon, that he's identified some cellular proteins that are involved in packaging things into exosomes and different messages, and he's done a wonderful job there. So we think that although it's not involved in envelopment, the escorts are exosomes that are made via the escorts are sending out messages that help prepare neighboring cells.
That's very cool. So you want to know what it's sending out, right?
Yeah, what is it sending out? How is it sending it out? How are from a virus perspective, how is it getting selected to get put in the exosomes? And he's got some good insight into that.
So that's another example of following your curiosity. It's going to lead you somewhere totally different, but that's the way it should be.
And once his paper came out, we had the discussion: do we follow up this exosome hypothesis, or do we figure out envelopment? And I would have preferred him to go the envelopment route and look at some other alternative envelopment things. But I felt he had earned the right to do what he wanted to do. He wanted to do exosomes, and he's done a great job, and there'll be a great story hopefully coming out about that.
But certainly some part of Escort is involved in envelopment, right? Maybe not Escort 3.
Well, I think it's probably more like influenza, where it doesn't.
Oh, it's independent. Okay.
And that's so there was a viral protein that was knocked out by a group in Germany, Jens von Eidem, and they actually have these stalled bud necks. So you have the virus particle attached to membrane via a small bud neck. Yeah. And that protein is UL71. And so our hypothesis was that UL71 was working with the escorts.
Right.
Now I think it's probably working independently, and I'd love to know how, and Jens would love to know how, and I think the field in general would love to know how. And so that's kind of, we're looking at different ways that is.
So remind me, how does CMV maturation occur? So a capsid forms in the nucleus?
In the nucleus.
So then what happens? It buds out of the nucleus?
So it goes under this, or it buds into the nuclear space and then into the cytoplasm. So kind of like an envelopment, de-envelopment together.
Right.
Nucleus.
Like herpes simplex.
Like herpes simplex.
Right.
Yeah. And once it gets into the nucleus, we believe it encounters that assembly compartment where you have its tegument proteins. It can acquire its tegument, and then it gets a membrane to get its final envelope. And there's a lot of different hypotheses for what's the source of the membrane, but it'll get its membrane, get enveloped, and then egress out of the cell from the assembly compartment region.
It takes the secretory pathway out, right?
Yeah, it goes out, yeah.
That's how it
and you can see them in multivesicular bodies, so it could, sometimes is individual particles that egress out, and also sometimes is like an exosome type thing where the multivesicular body might fuse.
So your idea is that all of this is independent of Escort machinery?
Our data would suggest that it is.
It's the same for simplex. Have people looked at it there?
Simplex requires the escorts.
It does.
Yeah, and we repeated that as part of our study. Yeah.
Is there a signal in a herpes viral protein? Like these retroviruses have a late domain, PPX something or other, right?
They vary, yep.
And do you see that in simplex?
You can find late domains all throughout CMV proteins as well.
Really?
But
if you take them out, it doesn't matter?
So the 2009 paper that I alluded to, I believe it was that paper, they actually mutated some of the domains, and I believe they didn't find any difference. We personally haven't done anything with those late domains.
Okay. All right. So you're following up what's in these vesicles, right?
Yeah.
What else is going on that you're excited about?
Oh, we've got a lot of things in the lab we're excited about. So we've been studying one protein that we knocked out in fibroblasts. You know, fibroblasts is what we use for CMV. It grows a high titer. And we've knocked it out and there was no phenotype. I know at least 3 or 4 other labs that have knocked it out with no phenotypes. And so it's kind of always been a protein that's kind of been set aside. But we found we've been switching from using fibroblasts to using these cells that are from a monocyte origin. There's different ones. Our one that we like are Kasumi 3 cells. Ian Murphy and Christine O'Connor piloted the use of these cells for CMV and knocked out the protein and found we make no virus particles when it's gone. And so, you know, fibroblasts have been great and we've learned a lot about them, but, you know, sometimes they don't tell the whole story. And you know, monocytes, of course, are the relevant, they're what they form latency in the host, the human host. So we're excited at following that up, and we've got some ideas on what's that doing, but I'm not quite yet ready to share those ideas because they might be wrong. But
no problem.
But we've followed up. We have some hypotheses there, so we're really excited about that protein and understanding what that's doing. Being interested in trafficking, we've got projects in the lab that are looking at some very important plasma membrane proteins are downregulated and understanding how that occurs and what's going on there. And we found an ubiquitin ligase that is not normally expressed in fibroblasts, and the virus actually induces it, so we can find it in fibroblasts, and it's leading to the degradation of some plasma membrane proteins. So we're following up, how does that get induced and what it's doing. So we're excited about that, and of course we're excited about UL71, which we think is doing development, and that's where our funding comes from. Our R01 is on development. So
is it fair to say you're moving away from Escort?
Well, we still have the Escort exosome project, but after that, I either have to switch viruses or move away from Escorts. Because
I think that's interesting, because now you're mostly focusing on HCMV.
Yeah.
I mean, the exosome project really is what's in them, not how they get out. That's known, right? So that's curious, because you wanted to combine the two, but now you found that they don't need each other.
They don't, but I think, combining them got me a job and it got me funded.
Yeah, of course.
It worked, I guess, in the end.
But that's how science is. You can never, and in fact, when you first said to your student, just do this and you'll find it's got a role in development, that you can't do that, right? Because you can't predict. You never know where things are. That's the beauty of science. In fact, that's the beauty
That's
of
what
it.
makes it fun.
You bet.
That's what you said. You never know what you're going to find, and when you do find that odd observation, you get really excited.
Okay. All right, speaking of fun.
Understanding it is kind of the fun part, right? Until you, it's miserable until you understand it, but yeah, it's definitely exciting.
Speaking of fun, let's wrap up with some fun questions here. So let me ask you, Leslie, if you hadn't been a scientist, what would you have done?
Well, when I was young, I wanted to be either a ballerina or a pilot. I know they're a little bit different from one another, but I had, different experiences. Yeah. But now, if I had to choose, I think it'd be really exciting to travel a lot and be a photographer. So I would have to say a National Geographic photographer would be what I'd like to do.
Cool. Do you like taking pictures?
Yeah. I'm not that great at it, but so I probably could never have that job.
Yeah, well, you could get good at it. So have you read David Quammen's book at all?
I haven't.
He's a science writer who works for National Geographic, and they send him all over with photographers to photograph for his articles. So that's pretty cool as well.
Yeah, I have a nephew who's doing that, so I'm kind of living life through him. So.
Yeah. How about you? What would you have done?
The boring answers, I probably would have gone into business, just got an MBA or something. Not as cool as going around the world photographing things. But, I always liked garbage men growing up. Maybe I would have been a sanitation worker.
And we have some listening to the podcast, They've written in.
We had bunk beds in the bedroom, and I used to climb up on them and pretend like I was throwing trash in the back and then jump off. It was corny, but that was one of the things I did growing up.
Now, Leslie, what do you think is your most important contribution to the field?
Well, I guess it's, the core work that we do is really trying to understand the nuclear trafficking of GAG proteins and what they're doing. And, if we're able to really convince ourselves that there is this packaging pathway that occurs in the nucleus, at least for RSV, I think that would be a very different paradigm to understand RNA packaging. So that would be, if we can nail that down, we'll be very excited.
Are you going to work forever?
No. I don't know. No, I mean, I don't think I'll work forever. I think that science is the kind of thing that you hate to ever stop because there's always another discovery to be made, and it's kind of just, you know, it just pulls you in. So hopefully I'll know when the right time is to stop. I don't know when that's going to be, but I'm sure I won't. I'm sure I won't. Although my father's still working in his 80s, so you never know.
How about you? What's your? I know you're young, but what's your most important contribution to the field?
I think this last paper was important because it kind of cleared up a little bit of discrepancy in the literature and helped give clear understanding to this process. So.
Okay. I'm not going to ask you if you're going to work forever because you're too young.
If my Powerball ticket had one left,
you wouldn't be working now.
I think you would. Actually, I think you would. If you won that, you would use it for your lab, probably, right? That's what I would do.
Yeah, I would go to the website and donate to TWiV. What's the website again?
Microbe.tv/contribute. Yeah, all different ways you can do that. Yeah. Have you read an inspiring book lately?
I knew you were going to ask that question. So I guess the book that I'm in the middle of that I'm thinking a lot about is Grit. So Angela Duckworth is a psychology professor at UPenn, and one of my daughters got me into that book. And so I think it's really interesting. I think the whole question that we're facing, certainly in medicine and probably in other careers, is this idea of people getting burned out. So burnout, I think it's a big problem. So trying to understand how to be resilient in the current environment, I think, is really important. So we're actually working with our MD PhD students through a year-long seminar series that we're working on, trying to promote resilience, wellness, understanding grit. What are the qualities that we really need as scientists and as physicians to really continue to work forever, maybe, is the answer, right? So if everyone really, we love what we do, but there are things that can sometimes take away from that joy of living. So I'd say Grit is inspiring me at the moment.
Grit. How about you, Nick?
Yeah, so I read a book a couple years ago, and I believe it's called Scientists. I don't know who wrote it. I can email you that, but it was essentially a history of science, and it talked about some of the major scientists. And you asked me what was inspiring, and to me it was inspiring because for many centuries, science was done as a hobby.
Yeah.
And I get paid to do it. And that's a crime. Like, it's fun, and I enjoy it, and I enjoy coming to work every day. And so I like that book because it kind of reminded me that what I do every day is a privilege.
It is.
That used to be a hobby for the rich people. But.
For wealthy people. So Darwin was a wealthy guy. That's why he could do what he did, travel to places and catalog animals. And it was actually, science was a part of philosophy originally, right? That's why we get a doctor of philosophy still to this day.
True.
All right, one more. This is easy. DNA or RNA?
RNA.
DNA.
I stole that from Goggles. What's the name of the podcast? Goggles what? Goggles Optional. Goggles Optional is a podcast done by Stanford scientists. And they interviewed me a couple of weeks ago, and they did these fast questions. And that's one of them.
They say it's an RNA world now? Sorry.
Well, he didn't give me the real option. DNA, RNA, or protein. I would have gone protein.
Oh, okay.
You would have done protein?
Proteins are the one that pack their bags and go to work and get the work done.
Yes, I'll add it for the next time. DNA or RNA or protein. Just like living things, TWiV evolves too. We can do different things. All right. That is TWiV 518. You can find it at Apple Podcasts, microbe.tv/TWiV. And most of you listen on a phone or some kind of mobile device, use an app. Please just subscribe to us so you get every episode. It really helps us to have subscription numbers. And I know a lot of people just listen now and then without subscribing, but it will help us if you do subscribe. So please do that. It's free. It doesn't cost you anything. One person told me once, I like to control what goes on my phone. And I said, why? Who cares? You got plenty of storage. Why would you care about that? So subscribe. Send your questions and comments to TWiV at microbe.tv. We usually read them in every episode, so we really appreciate that. And consider supporting us. If you go to microbe.tv/contribute, We have many ways that you can help us out, including we have a Patreon account. And if you can give us $1 a month, that would really help us for our production costs and really to do more. We have lots of ideas for other podcasts and shows, and we need some support to be able to do that. So please, microbe.tv/contribute. And while you're at it, go to youtube.com/PROFVRR and subscribe to my YouTube channel. There's always something we ask you for every week. I'm sorry about that, but I think in exchange we give you interesting stuff. My guests today have been from the Milton Hershey Medical Center, or College of Medicine in Hershey, Pennsylvania. Leslie Parent, thank you so much
thank you,
for joining.
Vincent.
Really appreciate it. And Nicholas Bukovic, thank you so much. What's the origin of that name?
Right near the Ukraine-Polish border.
Cool.
Yeah.
Neat.
If you look in the maps over time, it often changed what country it was in. But
Neat.
it's in Ukraine today.
I just visited a social security office. You can tell I'm old. I'm 65 or older. And they had a sign with English, Spanish, and Polish, and some Cyrillic language that I didn't understand. So we have a lot of, where I live, there are a lot of Polish communities. So I think that's pretty cool. And that's what's great about this country, right? This great diversity of people that contribute to all the things that we do. I'm Vincent Racaniello. You can find me at virology.ws. I want to thank the Biomedical Sciences Program and the Department of Microbiology and Immunology for hosting me, and Praneet for helping out. You answered a lot of emails and did all the arranging. Thank you. I know it's hard to do. It's a pain in the neck. So thank you very much for doing that. And our little audience here, thank you for coming. I want to thank the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for his music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral. Thank you.